So the topic that we are having, that is basically the enzyme inhibition. Okay. So what is enzyme inhibition? Inhibition means the any like the molecules which is stopping the enzyme. You can say that the reaction. Stop the reaction or the product is not formed. Okay. So the product that is basically attached with the enzyme and the product is not formed in actually, so this is what this is the you can say that the molecule that is stopping the enzyme activity. So the inhibitors are basically that stops the enzyme activity in our body, activity in body, that is called the enzyme inhibition. Okay. So the molecules that are like, you can say that they are not like converted into the products, so the molecules that are called the enzyme inhibitor, and if the process is there, so we are calling that an enzyme inhibition. Okay. So we are having many like the products, we are having the molecule that act as inhibitor. So we are having the molecules, we are having the cyanides. Cyanides is basically the poisonous substance which basically like you can say that stops the, it is a poison substance which stop the ATP production of our in our body. So there the ATP production will be stopped, so how we can survive? So this is basically dangerous for our body too. We are having many like drugs we are having, that is penicillin, sulfa drugs, they are metabolites many. Okay. Penicillin. Okay. Drugs, we are having sulfa drugs, and we are having the antibiotics, poisons, we are having many inhibitors which basically stop the enzyme activity. Okay. So if we talk about the inhibitors, so we are having two type of inhibitor. Okay. So first one we are having, that is basically a, we are calling that as a competitive competitive inhibitor. Okay, what does it mean? It means that if for example there is a active site, okay, there is active site and a molecule, this is basically what this is the active site. Okay, this is active site and the substrate which is like this is the enzyme, for example any enzyme, as you guys all know, every enzyme is having the specific substrate. Okay. So the substrate which is attaching with it, it is not that one substrate, that it basically the substrate which is attached to the enzyme. So we are having the succinate molecule. Okay, we are having a succinate molecule, and for that we are having the enzyme. If you talk about, so we are having succinate dehydrogenase. Okay, this is basically enzyme. Okay, succinate dehydrogenase. Okay. So a molecule that is succinate which is attached, which is like attached to this one enzyme. But likewise there is a enzyme, there is a molecule which is malonate malonate, this is basically similar to succinate. Okay. So the succinate is not joined, the malonate is basically joined with this. So the, you can say that the product will not form because this the enzyme of this, the enzyme is basically for this substrate, not for this, but this is unfortunately attached. So for from that, you can say that the enzyme activity will stop, activity will get stop. Okay. So this is temporary reaction. Okay. So we are also calling that as a reversible inhibitor. When this malonate detach from this, so the reaction will basically start, so the product will form, so that is called reversible inhibition too. Reversible inhibition. Okay. So this was about the competitive inhibitor which basically stop the enzyme activity, and there will be like you can say that there are many antibiotics, there are many like things, competitive enzyme you can use as drug. Okay. So there are many importance too. So if we talk about the next inhibitor we are having, that is non-competitive. Okay, non-competitive. So what is non-competitive? Non-competitive inhibitor, for example, this is the S active site. Okay, so the this is the active site. Okay, so the inhibitor, which is a molecule, for example, this is a molecule, this is not attached with this side, this active site, through which basically the substrate basically bind, but it is attaching with this side. It is like this, it is attaching with this side. So this is called the allosteric site. This is called the allosteric site, where the, you can say, the in the binding site of enzyme. This is the binding site. Okay, so this is attached with this. So this is the, you can say, the allosteric site. Okay, so this will basically inactive the enzyme. This will inactive the enzyme. So through this the enzyme property will, you can say, that stop, so the reaction will not occur. Okay, so this is as a temporary base, we are calling that as a reversible inhibition too. But what if this will, like, this molecule will denature this active site, so we are also calling that as a irreversible inhibition, which means that we are having two inhibition in this both. But in competitor we were having the inhibition, but in non-competitor we are having a reversible and an irreversible both. Okay, so reversible we are having that, like that it will stop the you can say that reaction or enzyme activity temporarily. But what if it is denaturing the enzyme, you can say enzyme active site, so that will be calling as the irreversible inhibition. Okay, so you can say that this is the, so we are calling this allosteric site where the enzyme inhibitor work not preventing by the formation of the enzyme, so we are calling that as a enzyme substrate complex. Okay, so through this the product is basically not formed, this is a complex substance. Okay, so after that we are having many, like non-competitive enzyme inhibitor that destroy the enzyme by, they will change the shape or they will denature the active site. So we are many, like ions we are having, that is, you can say that the mercury, we are having mercury. So mercury, silver, they basically what, they basically stop the enzyme activity, through which we are calling that as a mercury is very harmful for our body. So this is what, this is a non-competitive inhibitor which basically denature the enzyme. Okay, we are having the many products like heavy metals, we are having, these are the heavy metals. Okay, so we are having the cyanides, we are also having a cyanide, that is basically a poison substance, which basically, you can say that this is the basically the molecule which is very poison in nature, and they basically stop the ATP production. So through this the enzyme will not work, or the energy will not produce like in our body. So through this is basically harmful for our body. So the non-competitive inhibitor are very harmful, and this is like for temporary wise, so it is not at much harmful molecules. But these molecules which are attaching with the active side, so and not active side but from the sides one, so we are calling the allosteric side. Through this the enzyme can be denatured too. So these are very, like, both are very like harmful, but this is as much harmful, this is too much harmful. So this was about the enzyme inhibitor. In the next video we will discuss about the feedback inhibition.